Today, let's take a look at and get a basic understanding and introduction to the biophysics of ligand-receptor interaction. Just as some background information, let's look at some key points about cellular biophysics. First off, we know that cells are largely dynamic structures with numerous components. As such, they consist of a vast amount of interacting molecules. For example, the ions floating around in the aqueous cytoplasm, the DNA molecules within the nucleus, along with the various transcription factors and DNA and RNA polymerases, and of course the copious number of proteins folding, translocating, and undergoing conformational changes. This is a difficult concept to keep in mind as there are really too many moving components to mentally envision. One important point is, however, that the status of the cell is a constant struggle to reach both mechanical and chemical equilibrium. In this case, the equilibrium can be seen as a result of the desire to minimize free energy and maximize entropy, which aligns with the laws of thermodynamics. As such, we may be interested in the probability of a certain status of the cell existing. To move into a mathematical approach to the status of cells, statistical mechanics can be applied to compute the probability of all the different microscopic arrangements of the cell. These microscopic arrangements are also known as microstates. In the ever-changing cellular environment, there exists a plethora of microstates. For example, the interaction between a ligand and its receptor represents a series of microstates or arrangements of the ligand around the receptor. Based on this, a probabilistic approach can be used to calculate the average behavior of a system. Otherwise stated, what are the chances of a ligand binding to its receptor? Just as a side note, this is why an elevation of certain ligands is important for proper function. For example, a higher concentration of a given hormone will increase the likelihood that that hormone interacts with its receptor and exerts the action that is intended. This is why if hormone deficiencies occur, the effects of those hormones are not realized. It is not that a higher concentration of hormones means the effect is stronger. Instead, a higher concentration simply amounts to a greater chance of that interaction and effect occurring. Lastly, just to reiterate, a microstate again is one particular realization of the microscopic arrangement of the constituents for the problem of interest. Now, let's take a look at a visual and simplified model of this. This model here shows ligands in a solution in the presence of a receptor specific for those ligands. This simple figure introduces the idea of treating a solution as a lattice structure in which the positions that can be occupied by ligands are dictated by a discrete set of lattice sites. The microstates of the system correspond to the different ways of arranging the ligands among the provided lattice sites. Microstates 1, 2, and 3 represent situations where the receptor is unoccupied by the ligands, while microstate 4 here has the receptor occupied. Therefore, in microstates 1, 2, and 3, there are a number of ligands L distributed in the solution. In microstate 4, however, there is one ligand bound to the receptor, leaving L-1 ligands remaining to be distributed in the solution. This makes sense since this essentially is saying that of the total number of ligands given by L, one can be thought as of being taken out of solution as it is bound to the receptor, leaving the total number of ligands available in the solution missing one of the total. In this case, microstates 1, 2, and 3 provide 12 ligands near the receptor that are free in the solution. By contrast, microstate 4 shows 11 ligands free, while 1 is bound to the receptor. This of course corresponds to L ligands in the cytosol in microstates 1, 2, and 3, and L-1 ligands in the cytosol in microstate 4. Now, let's use this model and statistics to determine the number of different arrangements of the ligands in the cytosol with the receptor. Keep in mind that this also means we are determining the number of microstates. Essentially, we can use counting to determine the number of ways that we can arrange L ligands among the omega sites. The important factors here are that the ligands are all indistinguishable and only one ligand is allowed per site. When placing the first ligand, all of the sites are available or open. This means that if our total number of sites is equal to some number given by omega, we have omega choices to place the first ligand. Once the first ligand is placed, we now have one less spot available. This means that we have omega minus 1 spots left to place the remaining ligands. Of course, since one ligand has been placed, we now only need to place L minus 1 ligands. If we were to repeat this process, we end up with one less available spot each time we place a ligand. This means that initially we have some number of spots omega, then we have omega minus 1 spots, then omega minus 2, omega minus 3, and so on. This continues until we have placed all of our ligands. Here we have stopped at one term before all the ligands are placed, as shown by omega minus L minus 1. If we simplify this here by distributing the negative, we see that our last term here becomes omega minus L plus 1, meaning our last term says take the total number of sites omega and subtract the total number of ligands placed plus 1. This again essentially means that this last term represents a situation where we have placed all but one ligand. If we continue the sequence to the very last term, this of course gets us to omega minus L, meaning all ligands have been placed. Omega minus L minus 1, meaning all ligands have been placed and we have also taken away one additional spot, and of course our last three terms being 3, 2, and 1. The last three terms simply represent a situation that would occur if we kept subtracting more and more spots away from the total number. Eventually, we would end up with one spot available out of the total number omega. If we multiply and divide by this new term, let's take a look at the numerator and the denominator. In the numerator, we get this series shown here. This is basically saying that we start off with the total number of sites omega, and we subtract one additional site each time an object is placed until we reach one site remaining. This is interesting since if you take a look at this here, what is this? At a closer look, you'll see that this is a factorial. Therefore, the numerator of this term is simply equal to omega factorial. Now for the denominator. In the denominator, we have the last few terms of the sequence starting with the total number of sites minus the total number of ligands that need to be placed. This continues on to the total number of sites minus the number of ligands placed minus one additional site. Similarly, this continues until the final term 1 is reached, meaning only one site is left. Just as above, if we have some term omega minus L, and we subtract one additional site each time and multiply these numbers together, what is this? Well, this of course is equal to omega minus L factorial. So the numerator is omega factorial, and the denominator becomes omega minus L factorial, making the overall formula shown here. Now take a closer look at this. What is this? Hopefully you recognize that this is the permutations formula. This is saying take the total number of rearrangements and divide by the rearrangements of the items we are not interested in. In this case, the total number of sites divided by the sites available after placing L ligands. To finalize this, we know that our L ligands themselves can be rearranged. However, there is no concern for the different ways to rearrange these ligands because again, we are treating these ligands as indistinguishable. This means, for example, that if we have ligand 1 and ligand 2 placed in two specific spots, we do not care if their positions are swapped. From an assortment standpoint, as long as ligands are shown in a given orientation, this is all that really matters. Swapping the positions of those ligands does not matter and therefore should not be counted as a unique assortment. With this in mind, we need to also divide by the different arrangements of L ligands, being of course L factorial. By doing so, our prior formula becomes this one shown here, which you should also recognize as the combinations formula. And just as a brief visual of the prior points, here is a quick grid of a sample situation. Initially, we have no ligands present in this grid, and if you count the number of available spaces, you'll see that there are 20 open spots. This means that in this case, omega is equal to 20. If we were to place one ligand into this solution, there is now one space taken up, leaving us with 19 or omega minus 1 spots. Continuing to place ligands in this grid will result in one less space each time until we are left with only one space left, which is equal to, of course, omega factorial. Now again, if we have this arrangement shown here, all these ligands are identical. This means that if we were to swap any of these ligands around, but have them remain in the same overall arrangement, this assortment is identical to the last. This means that we are not concerned with the rearrangement of these ligands with the given orientation. Because of this, we divide by the arrangement of the ligands, or L factorial. This gives us the number of unique arrangements possible in a solution with omega available spots if we choose some number of ligands L.